Okay, so in a series of 4 videos we will be discussing blood flow and circulations, and there are 6 main topics that we have to cover in order to understand this topic pretty clearly. The first thing is we need to understand what local intrinsic control of blood flow means. Then we will move onto hormonal extrinsic control of blood flow, then coronary circulation on the heart, then we'll move into cerebral circulation in our brain, then we'll move with skeletal muscles, and finally we'll end with skin. Before we jump into the first topic of this video, which is local intrinsic control of blood flow, we need to understand that blood flow varies from one organ to another. Okay? Blood flow to an organ is regulated by altering the arterial resistance, and it also can be varied depending on the organ's metabolic demands, and we will see this theme over and over in this video. So let's go ahead and start with local intrinsic control of blood flow. And the thing that we need to realize is in intrinsic control, we have two things that we need to discuss, which is first examples of local control. What does local control mean? And then we'll go into the mechanism, which explains local control of blood flow. So let's start with some examples of local control. One of the first examples we want to talk about is autoregulation. So the blood flows to an organ. It remains about constant over a wide range of perfusion pressures. Organs which exhibit autoregulation are the heart, brain, and the kidney. So the autoregulation again happens in the heart, brain, and kidney. Okay, so let's give an example. So if perfusion pressure to the heart is suddenly decreased, that's not good. We need a vasodilatory compensatory mechanism, and that happens. So we have compensatory vasodilation of the arterioles, and that will maintain a constant flow. Okay, so that's point 1 here of autoregulation. Now let's move on to point 2 here of active hyperemia. Okay, blood flows to an organ is proportional to its metabolic activity. That's what this means. So let's give an example to understand this a little bit better. The active hyperemia example is if metabolic activity and skeletal muscle increases as a result of running or some kind of activity or exercise, the blood will flow to the muscle, will increase proportionally to meet that metabolic demand. Makes sense. We need more blood when we're exercising in order to take care of the lactate that we're building up in our muscles, in order to provide oxygen to the muscles, depending if we're an aerobic or anaerobic exercise. Now, the last thing that I want to talk here, or another example of local control, is reactive hyperemia. What does that mean? Okay, reactive hyperemia is an increase in blood flow to an organ that occurs after a period of occlusion of the flow. So if we occlude the flow, then we have something called reactive, so it reacts to the hyperemia. The longer the period of occlusion, the greater the increase in blood flow, which is above pre-occlusion levels. Excuse me. Okay, so far let's review what we talked about. We talked about local intrinsic control of blood flow. We gave 3 examples of autoregulation, active hyperemia, and reactive hyperemia. Now the second part is this local control of blood flow is let's understand the mechanism which explains local control of blood flow. There is 2 hypotheses that we need to mention. The first one is the Myogenic Hypothesis. The second one is the Metabolic Hypothesis to explain this local control of blood flow. So in the Myogenic Hypothesis, this hypothesis explains autoregulation but not active or reactive hyperemia. Okay, so this hypothesis is good, but it only explains autoregulation. Okay, it's based on the observation that vascular smooth muscle contracts when it's stretched. Okay, let me give you an example. If perfusion pressure to an organ suddenly increases, okay, the arterial smooth muscle will be stretched and will contract. The resulting vasoconstriction will maintain a constant flow. Okay, without vasoconstriction, blood flow would increase as a result of increased pressure. Again, without vasoconstriction, blood flow would increase as a result of the increased pressure. Okay, so this is the myogenic hypothesis. Again, explains only autoregulation, but does not explain the active or reactive hyperemia, which we talked about in point 1. Okay, so now the metabolic hypothesis. Okay, this metabolic hypothesis is based on the observation that tissue supply of O2 is matched to tissue demand for O2. Okay, we have vasodilator metabolites, which are produced as a result of metabolic activity in tissue. These are listed here, such as CO2. We have protons or hydrogen ions, potassium, lactate, and adenosine. And so let's give an example of hyperemia. So this takes care. So this hypothesis addresses active hyperemia. So let's give the first example. If the metabolic activity of a tissue increases, so let's say during an exercise, both the demand for O2 and the production of the vasodilator metabolite increases. Okay, so here is the two criteria. And during exercise, we have demand of O2 increase and also vasodilation. So the increased metabolite causes arterial vasodilation, increased blood flow, and increased O2 delivery to the tissue to meet demand. So when we're exercising, the metabolic hypothesis explains this by we need more oxygen and also we need vasodilator metabolite, which is lactate in this case. Let me give you another example and we'll end with this for this subject. And it's if the blood flow, let's say, to an organ suddenly increases as a result of a spontaneous increase in arterial pressure, then more O2 is provided for the metabolic activity, okay? But at the same time, the increased flow washes out vasodilator metabolites. As a result of this washout, okay, arterial vasoconstriction occurs, resistance increases, and blood flow is decreased to normal, okay? So this covers the local intrinsic control of blood flow. Next video, I'll be talking about hormonal extrinsic control of blood flow. I appreciate your feedback.